Hi everyone, I'm going to talk about the 3 phases of gastric secretions here. What does that mean? What's a secretion? Something that the cell sends out, right? And what's really neat about this is we're going to be talking about exocrine secretions and endocrine secretions. Did we learn about these hormones? Well, the endocrine chapter briefly touched on them. But these are hormones that are going to be staying within the same organ. There are a few that are going to from the intestine to the stomach, but a lot of these are staying within the same organ. So are they hormones? Is this hormonal, or is this autocrine or paracrine? We're going to be talking about paracrine hormones here. And gastric, what does that mean? Relating to the stomach. When you think of stomach, what do you think of? You think of stomach acid, right? And if you've been reviewing your types of cells, you know that we need acid to activate our enzymes, which cleave the proteins. All right? So we'll discuss that again briefly here. All right, so the first phase, when you first find yourself with food, or think of food, or even smell food, that's the first phase, before it even enters your stomach. All right? The cephalic phase. Cephalic is in your head. You're thinking about it. Sight, smell, thought, even taste, but before it gets to your stomach. Activates your vagus nerve, cranial nerve number 10, which activates your gastric diffuse neuroendocrine cells. Let me write that out for you. You've been talking about that with the cell types, right? But let me write it out. Diffuse neuroendocrine DNES cells. Diffuse neuroendocrine cells. Alright. So the gastric diffuse neuroendocrine cells then promote gastrin, promote histamine, and inhibit somatostatin. Now the whole goal is to increase hydrogen ions, right? We have these parietal cells, if you'll remember, and they're secreting hydrogen ions. Now, if we want to promote that secretion, so we're going to stimulate. We have stimulation from gastrin and from histamine. Somatostatin is inhibitory. Well, look, we're inhibiting the inhibitor. So what happens? We turn off that inhibition. Oh, you know what? There's a glare. Let me take care of that glare. All right. Is that better? That's much better. All right. See here? It's inhibitory, but we inhibit the inhibitor. So that turns off that inhibition. All right. So the cephalic phase, before food enters the stomach. So it's not even there, but the stomach is preparing by making hydrogen ions, by secreting more hydrogen ions. Does that make sense? You've got your vagus involved. Of course, you have your vagus involved all throughout, but gastric diffuse neuroendocrine cells. Gastrin, histamine, somatostatin. All right, it's going to be the same down here. Some of the same players. But now we have food in our stomach. In the gastric phase, which makes sense, gastric, stomach. Now there is food in the stomach. So what happens when the food goes into the stomach? It distends the stomach. It stretches it, distends, like stretches. So food in the stomach that that activates your vagus and your enteric nervous system. Remember that some call it the gut brain, the nerves within the nervous system, and those activate the gastric diffuse neuroendocrine cells. But look at this. What is this? Another way to activate the gastric diffuse neuroendocrine cells is to have partially digested proteins. That's really interesting, right? Because now you actually have food in the stomach. The stomach can be digesting them, and as it's digesting them, those proteins that are getting digested are activating this, saying, yeah, digest some more. Does that make sense? We'll talk some more about it. All right, we have gastrin, histamine, somatostatin. So do we want to activate or inhibit gastrin? We want to activate it, right? Because we want more. Same thing with histamine. We want more hydrogen ions. And we want to inhibit somatostatin, because somatostatin is the inhibitor. So we will now turn off that inhibition. We inhibit the inhibitor. But here's what's interesting. Hydrogen ion, right? You're going to have more pepsin. Hydrogen ion means more pepsin digesting proteins, which means more partially digested proteins. So let's write this out. So the more hydrogen ions you have, the more activated pepsin you have, the more proteins are getting digested, which means the more you're going to activate the gastric diffuse neuroendocrine cells. What kind of feedback is this? What is that positive feedback? So we have positive feedback going on. Isn't that exciting? So we're making hydrogen ions. The more food in the stomach, well, as there's food in the stomach, and the more you have partially digested proteins, the more hydrogen ion secretion you're going to be getting, which means more partially digested proteins, more hydrogen ions. Now, is that going to go on forever? Like, would the pH get very low? And it does get very low. But once it reaches a pH of 2, then you have somatostatin activated. So at a pH of 2, somatostatin will get activated and hydrogen ion secretion will decrease. Does that make sense? So for the gastric phase, the food is now in the stomach. You're activating the vagus and the enteric nervous system. The gastric diffuse neuroendocrine cells now are again going to activate gastrin and histamine and inhibit somatostatin, which is an inhibitor itself. So you're going to get more parietal cell activity and more hydrogen ion secretion. But there's another way, right? Partially digested proteins will also activate the gastric neuroendocrine cells, diffuse neuroendocrine cells, and activate gastrin. Histamine inhibits somatostatin, which will activate or promote hydrogen ion secretion by the parietal cells. Positive feedback there, right? The more hydrogen ions, the more pepsin activated to digest proteins. The more partially digested proteins you get, the more activation of gastric diffuse neuroendocrine cells. And it keeps going until when? pH of 2. Then somatostatin is activated and hydrogen ion levels secretion starts to decrease. All right. Now for another section, which is really kind of two different phases in this phase. We have, well, what about what happens after the stomach? Does it start releasing just a little bit into the intestine? And initially the intestine's like, yeah, let's have more. Or it's kind of like, keep it up, keep it up. So what happens is you get these partially digested proteins in the duodenum, the first part of the small intestine. That stimulates the vagus, which stimulates the intestinal diffuse neuroendocrine cells. Notice how up here it was gastric. Here now it's intestinal. And that's going to activate intestinal gastrin, which will get into the bloodstream and promote parietal cell hydrogen ion secretion. Because initially the intestines are like, yeah, keep it up. So initially, look at it, it's all green. It's go, go, go. It says more hydrogen ions. But as the pH in the duodenum starts to decrease and the lipid content starts to increase in the duodenum here, we have the entero, E-N-T-E-R-O, gastric reflex. By the way, guys, this, you can see this whole figure in your textbook. So if this is hard to see at all, or you want to follow along, you can find it right there in the textbook. Alright, your enterogastric reflex, look at that, is going to inhibit parietal cell activity. Your vagus here is now going to activate secretin. If you're having trouble remembering, what does it secrete now? Secretin. So what is secretin going to do? Now we're in the, ooh, wait, too much phase. Secretin is going to inhibit. And gastric inhibitory peptide, what do you think that does? So if you're having trouble remembering what inhibits the gastric parietal cells, gastric inhibitory peptide. All right? And they're all saying, no, parietal cells, enough, enough. Turn down that hydrogen ion secretion. Does that make sense? So initially the intestine says, yeah, more, keep it up. Right? Stimulates the vagus, which stimulates the intestinal diffuse neuroendocrine cells, which stimulates the intestinal gastrin, travels in the bloodstream, and promotes hydrogen ion secretion by the parietal cells. Now, after a little more, once the pH starts to lower and the lipids start to increase, you get the enterogastric reflex, which inhibits parietal cell activity. This is the duodenum saying, whoa, wait a second, enough, enough. And as enough being hydrogen ions. And it stimulates the vagus, which in this case, look at that, secretin, gastric inhibitory peptide are going to inhibit parietal cell activity. So these are the three phases of gastric secretions. Cephalic, gastric, and intestinal. I've definitely had essay questions on this. You definitely want certain words, and this test is definitely big on cell types. So if any of these cell types are confusing, I'd look them up, ask me questions, and hopefully all that made sense. It's a lot of information. I suggest writing it out and maybe like write this out and then a little paragraph underneath. Write the next one out and a little paragraph underneath. And then I think you'll really know it. And you know what the great thing is? Everybody eats food. And so think about it. Like, oh, hey, what phase is my stomach in? You know, what phase of gastric secretions am I probably in right now? If you pass by a bakery or something, right? And enjoy that smell, you're in the cephalic phase. Once you eat something from the bakery, you're in the gastric, and then later, the intestinal phase. All right? And look it up in your textbook, 22.12. Thanks, everyone.